Okay, perfect. Okay, well, thank you everyone for being here for my presentation. My name is Shradda Bhonsle, and I'm a fourth year CMDB major and an undergraduate researcher in Dr. Sladig's lab. So currently, Chancellor's Research Fellowship has been a really huge part of making this research happen. And I just kind of want to get started with introducing the title of my research, which is Identifying Alternately Spliced Genes in Metabolic Disease Pathways. Oh, sorry. Okay. All right, so just before we get started with actually talking about my research, I kind of wanted to explain what metabolic disease is and why do we care about researching it. So metabolic diseases, they are a growing problem in our society, and it will likely affect most of us at some point in our lives. According to the NIH, so as of 2022, nearly 1 in 3 US adults do meet the criteria for metabolic syndromes, and they include numerous numerous conditions, which include abdominal obesity, hypertension, type 2 diabetes, etc. So we are seeing that these problems are becoming increasingly prevalent for millions of people, and they're becoming a larger and larger source of morbidity for these people. Again, it's not just about the numbers, it is about obviously the impact that it has on the patient's life and as well as like premature death. And just developing some of these metabolic diseases can actually put you at risk for developing more serious chronic conditions on top of it. So this really makes it urgent to understand the pathways by which these metabolic diseases work and also identify how we can improve the current therapies. So while the current therapies on the market right now, they do manage symptoms, but I believe that we need to go beyond managing symptoms to actually understanding how the tightly controlled signaling pathways in our body get dysregulated and lead to us developing metabolic disease. Okay, so whenever we think of metabolism, the liver comes to mind. So we have to understand how the most important metabolic organ works to understand these diseases. Now, the liver is very crucial to maintaining the metabolic homeostasis in our bodies. It has tightly controlled signaling pathways. So when we're fasting for long periods of time, for example, I'm going to kind of refer to this diagram. When we're fasting for long periods of time, our blood sugar drops. So your body needs less insulin, and usually less insulin causes other hormones to activate this protein, which then activates a receptor that we call PPAR alpha. And this receptor specifically helps your liver produce glucose as well as produce ketone bodies to feed your brain and other organs when you haven't eaten for a while. And it's like a good amount of fasting. So in contrast, when we're in a fed state, so when we're eating, our liver will start storing excess nutrients because the insulin levels in our blood are higher. And so our glucose will begin being stored in the liver as glycogen. However, in people who are diabetic or resistant to insulin, for example, the glucose won't be captured into the liver, and instead all the extra insulin encourages fat to be stored in the liver, which leads to non-alcoholic fatty liver disease. So I just give these examples because it helps you understand how our liver manages fed and fasted states and the medical issues that can arise when these signaling pathways go wrong in the liver. So on the bottom of this graphic, you can also see HNF4 alpha. This is known as a master regulator of the liver. It's a transcription factor that I'm highlighting because it has a very major role in regulating all of these processes. Okay, so HNF4 alpha, what is it? So in the Slatic lab, we do study this highly conserved transcription factor. It stands for hepatocyte nuclear factor alpha. It's primarily expressed in the liver, and it's very essential for our liver to function properly. And this gene, just going a little in depth, so we see in the graphic that it has two promoter regions. It's labeled as P1 and P2. And these promoter regions can be expressed under different conditions. So on the right, in figure B, we do have HNF4 alpha exon swap mice. We call these alpha 7 homozygous mice because they differ at one promoter region. And these are exon swap mice that have been generated so that one, like the exon 1D replaces 1A, and so you have these two exon 1D promoters in that at those promoter regions. And so it is different compared to the wild type version of this gene. So this isoform of HNF4 alpha is actually not usually expressed in healthy normal adult livers, but it has been identified as being upregulated in liver cancer, as well as in the liver of mice that are fed high-fat diets, as well as in alcoholic livers. So that generally suggests that this isoform of HNF4 alpha has a role in metabolic dysregulation and disease. So in my research, I use the HNF4 alpha 7 expressing mice as a model for studying the mechanisms of metabolic disease. So the next question is just how is HNF4 alpha expression and other glucose and lipid metabolism genes, how are they regulated? So currently, a lot of studies, they are showing that alternative splicing is a major process in regulating how these genes are expressed, and specifically the genes that are involved in metabolic disease pathways. So they have shown that the genes that have been involved in these disease pathways have been shown to be heavily alternatively spliced. And so that's where I kind of want to go into what is alternative splicing and why are we focusing on it to learn more about the disease mechanisms. So it's a very common occurrence in our genome. Most of our genes with multiple exons undergo this process. It's essentially how we generate protein diversity. So we have like 25,000 genes, yet we're able to generate nearly 90,000 proteins and above. Now, the general principle to kind of understand is that with the central dogma, it's a very common dogma, right? So it's like DNA, and then you get all the introns spliced out, you get mRNA, and then we are able to produce different proteins based on how that splicing pattern occurred. So these splicing patterns are generating diversity, but occasionally they can result in pathogenic isoforms, and these pathogenic isoforms can contribute to metabolic diseases. So in this research, my main goal has been to identify where these real alternative splicing events take place, and that way we can use them as therapeutic targets. And primarily, I'm going to be doing this through analyzing RNA-seq data that we get from mice livers, but I'll go into that a little bit later. So in the liver, alternative splicing is very key to regulating the expression of HNF4 alpha, as well as other lipid and glucose metabolism genes, as I mentioned before. And previous studies have shown that any changes in the splicing patterns of HNF4 alpha can lead to dysregulation in the way that lipid metabolism genes are expressed, as well as contribute to metabolic diseases. And so it does suggest that there's a role for splicing of HNF4 alpha in liver specific, in how it regulates the liver specific genes in these disease pathways. Okay, so next I'm kind of going to go into what my hypothesis and the questions that I've explored for this research project have been. So some of the questions that I've explored are, does the mice being in fed versus fasted state, does that impact splicing events? As well as does the type of HNF4 alpha isoform impact splicing? And then primarily I'm trying to identify which alternatively spliced genes are implicated in the metabolic disease pathway. So my hypothesis is that fed and fasted livers will exhibit alternative splicing, and that wild type and alpha 7 homozygous livers will also express alternative splicing or exhibit alternative splicing. Okay, so next, my first step was actually in our lab, we did generate transcriptomics data. We used young adult, so 16 to 20 week old male mice, and we used them wild type mice as well as the exon swap mice. They were fed standard lab chow, and they were fed or fasted for 12 hours, so from 10:30 p.m. in the night to 10:30 a.m. in the morning, as well as, and then we harvested their livers. So then once we took these livers, we were able to analyze these livers for differential expression. So basically looking at these livers and inputting them into the RNA-seq data into these programs, so specifically DE-seq2 as well as DEC-seq, as I listed on the slide. And these two programs can kind of identify whether there's differential expression events going on or alternative splicing events going on. The programs were able to analyze these two data sets. So we did multiple replicates. They were able to analyze these data sets and basically narrow it down to 177 genes that it recognized as being both alternatively spliced as well as alternatively expressed. And so that really encouraged us to believe that there was a high probability that there were real splicing events taking place at least a couple of these 177 genes. All right, so the next step was manual curation. So I had to go in and basically look at the 177 genes that were called by the programs and manually curate it. And I was able to develop a inclusion and exclusion criteria based on how I would look at these genes and the kinds of, so I would compare them with various maps that I'll show right after. And I would basically was able to develop this criteria, and any genes that met all the inclusion criteria were likely alternatively spliced genes, and I could confirm that with the program, but any genes that met the exclusion criteria were likely not real splicing events. Now, the next slide I'm going to show you here is an example of a manual curation that I did for a gene. Now, this is a gene that did fall within the exclusion criteria, but I did kind of want to go through how I was able to exclude it out. Excuse me. So at the top, you see there's a gene map. It's actually the gene body, an RBP4 gene, and it's on UCSC Genome Browser. So I was able to actually compare this gene body with maps at the bottom, which are generated by those two programs that I was using. And those maps at the bottom were generated by DexSeq. So at the map at the top with UCSC Genome Browser, you see red bars and you see blue bars. Now, those are actually two RNA-seq data sets. So there's one that's fasted, represents males that were fasted, alpha-7 males, and wild-type males that were fasted, respectively, red and blue. So I was basically comparing at these exons, which you see as like those blue blocks on the gene body, how did the RNA-seq expression look at those two exons in these two mice models? And I was actually comparing how the expression looked with the maps at the bottom. So if you see, there are pink bars that are highlighted and circled. And those pink bars were actually the same. They were highlighted by the program in both data sets. So that meant that might have been an exon that was worth looking at. And so I was trying to match that exon bar to the UCSC Genome Browser gene body. In this case, it did meet a disqualifying criteria and thus was not alternatively spliced. That led me to believe, through my criteria at least, that it was not alternatively spliced. But this is technically how I analyzed each of those 177 genes. Now, once I finished going through and comparing all those maps for all 177 genes that the program called, I did conclude that all of the genes did end up falling into the disqualifying criteria that I developed. Out of 177 genes, though, there were 16 genes that weren't fully disqualified, so they didn't meet all the disqualifying criteria, but they also did not meet any of my inclusion criteria, and so I could not confidently label it a splicing target, but it would be something to look forward in future research. And although, I did get this result, these genes were still seen by the program to have differential expression, and so they may still play a role in liver dysregulation and the disease pathways, but just not something that my criteria was able to identify. So in the fasted mice, I also, the results also did identify a number of genes that were related to mitochondrial function as potential splice sites. So these mitochondrial genes are well known to be expressed during fasting conditions, and generally when we lose the function of those genes, so when they undergo loss of function, people can develop diseases like obesity, hyperglycemia, which is like high blood sugar, and hypertension. Overall, so the DexSeq was able to call over the 3,000 differentially spliced and expressed genes. It was able to narrow it down through the comparative data sets into 177 differentially expressed genes. And so once I developed that manual criteria, not all of them seem to be worth following up with wet bench testing. However, the 16 that were observed to be like mitochondrial related would definitely be worth following up on. And the livers, some of the notes that I did make while doing this research, the livers were harvested at the same time of day following the same fasting regime. So any differences that I believe, like I saw in the maps when I was comparing them, could have been due to any RNA-seq noise or any differences in the background of the mice. So I believe that like in the future, when we are able to come up with more accurate RNA-seq programs, it could more accurately analyze those gene targets. So with the mitochondrial genes that were implicated in disease, I think further research could definitely analyze the role of those mitochondrial genes in similar disease models, mice models. Another more relevant implication of the research is intermittent fasting. So, intermittent fasting has been something that's been going around for a while. It's becoming a really common way to improve liver health in the patients through diet, patients with metabolic disease. And so, previous studies, we've seen that intermittent fasting can alter HNF4 alpha activity. And so, studying its role and that if it's isoforms on the liver could definitely clarify like how it is able to regulate metabolism during the periods of fasting and feeding that you undergo in intermittent fasting. So this way it could definitely provide a lot of information for developing new diet interventions and addressing metabolic disease and liver health overall. So hopefully it can further preventative nutrition or even become a target for future drug therapies. So I just wanted to thank Dr. Sladek, Dr. Jose Lomelli, and Dr. Sarah Roddy, Dr. Jonathan Deans, and members of the Sladek Lab who have supported me in this, as well as the Chancellor's Research Fellowship and the UCR Research Symposium for making this happen. So thank you. Any questions?
Okay, if somebody has questions, please put them in the chat. Shradda, you can stop recording at this time.
Okay.
Yep. Or if you can unmute,